It's my pleasure to introduce our first speaker of the day, Romain Alderweireldt. I recently met Romain and his partner Ludivine at the Marfan European Network Conference in Vienna, where he presented their research project, which looks at protective modifier genes for the cardiovascular implications of Marfan syndrome. This project was inspired by Romain and Ludivine's young son, who has Marfan, along with the maverick spirit of PEARS pioneer Tal Golesworthy. Welcome, Romain, who's tuning in glamorously from Belgium.
Thank you very much. Thank you for the invitation, and thank you for the honor of being able to speak today at this webinar that I used to follow. So, Victoria, as you explained, our story with the 101 Genome Foundation begins with a diagnostic odyssey and a discovery. So we have 3 children, and our last child is Aurélien. And when Aurélien was born, it appeared quite fast that he was different, that something was different with Aurélien. And so we begin this odyssey. And at the end of this odyssey, we discovered that Aurélien has Marfan. And he has the Marfan syndrome, and for us it was really new because nobody in the family has Marfan. It happened, it's a de novo mutation that it's called. And for us it was really new to discover this world of the Marfan syndrome. And we heard that unfortunately Aurélien has what's called a neonatal form of Marfan, an early onset form of the disease, meaning that he has almost every symptom really, really fast in his life. So after discovering Marfan syndrome, we discovered that within this specific category of a neonatal form of Marfan syndrome, the life expectancy was really poor, with only a statistical life expectancy of a few months. And we understood that it was a terrible shock for us. And we went to the geneticist who helped us to get the diagnostic of Aurélien. This is this person there that you see on the screen. It's Professor Genomes. And Genomes helped us to understand that it's only statistics, that we are on the verge of a real technological revolution with the fact that now we are not working anymore only with a gene per gene approach, but we are really working with panels approach and exomes approach and even whole genome approach with the new generation sequencer. And it helps to understand the interaction between the gene, and that could sometimes have an impact. And we are lawyers, my wife and I, and so we spend a lot of time to teach us and to answer really, really different questions. And during this time, I discovered this study called the Residence Project. And in this study, a group of researchers had access to 600,000 genomes that they decided to reanalyze. And when doing this reanalyze, they identify pathogenic variant in this cohort that should have caused rare disease at a very young age. And apparently those people were not sick. At least 30 people should have been sick and were not sick. So with this discovery, with this study, I went back to Professor Genomes, and I asked him to explain me what was happening with those patients. And he told me about the role of the modifier genes, the protective genes, that could sometimes counteract the pathogenic effect of a gene. And with that, I decided to use the database funded by the Marfan Association, UMD FBN1, that gather all the pathogenic variants known as of today, and to cross those pathogenic variants with a database of control people called GNOMAD, where you find genomic information about people who are supposed to be in relatively healthy condition. And doing that, I found an intersection, and I went back to Guillaume Smith with this intersection, and he told me, yes, it's interesting, but what will be really interesting is to find pathogenic mutation that are in the same area that is known to be causal for the neonatal form of the Marfan syndrome, meaning the same form as the one that my son has. And so I went back to my study in my data and I identified in this intersection 24 individuals who are 24 variants who are at the same time classified as causative for the disease in the UMDF-BN1 and that you found in the regular population. So with this disease, with this observation, Guillaume thought that there is something to investigate further. And just for the story, years later, there was a publication that came with the same observation. And so with this observation, we have the hint that maybe somewhere in the genomes, there are other genes, protective genes, that could counteract the main effect of the deleterious effect of a variant in FBN1 gene. And with that, we were indeed inspired by the example of Tal Golesworthy. And since my son was born, it's followed by Dr. Philippe Rega in UZ Leuven, who's the person who was able to place PEARS in Belgium. And so we were following the progress of the PEARS and the work of Tal Golesworthy. And at one point, we realized that if some people like him is able to move the world and change the world, we should give it a try also. And so it's why with Ludivine, we created the 101 Genome Foundation, because we understood that there was a missing link between the patient association and the researcher. And we wanted to help the patient to participate to research and full research and allow the researchers to have access to the data that they need to try to identify the protective genes. With that, we created the first step that we followed. It's that we create a specific fund at the end of which we can find Professor Anne de Pape, which is really well known in the world of Marfan. And we opened this fund at the King Baudouin Foundation that allowed us to have tax deductibility in Belgium, of course, but actually all over the world, which is really a powerful tool. And we wrote a white paper describing this idea to go for the quest of the modifier gene. And with this white paper, we went to different scientists and we created our scientific committee to lead our research. And so those are familiar faces for the people involved in the world of Marfan. We have Jules de Backer, Bart Loeys, and Guillaume Jondeau, Catherine Boileau, and of course Anne de Pape, and Guillaume Smith, who's perhaps less known in this universe, but who really is leading our. And with that, we decided to design the GEMS study, which is currently led by Professor Paul Cook from Ghent University and Professor Bart Loeys from Antwerp University. And the main idea in GEMS is to try to identify patients with the same causative pathogenic mutation and identify patients who are at an advanced age unaffected carrier of the mutation, and on the other end of the spectrum, people who are affected severely affected at a very young age. So this is really the idea of the design of the study. In order to allow the study to exist, to be conducted, we decided with the 101 Genome Foundation to open our own biobank. So we create our own 101 biobank. And on the top of it, we create what we call our own biobank, which is a genomic cloud where we gather the DNA digitalized. So the idea is that you gather the biological DNA in the biobank and you transform the biological DNA into digital data that is stored in our genomic cloud. We created this genomic cloud, which is one of the first of its kind, with the help of Geraldine van der Roara, who was at the MIT at the time, and Dr. Colby T. Ford, and both of them, they wrote these books on the genomics in the cloud approach. So with that, we needed to go back to the patient. And here, as a patient, we had this experience that perhaps some of you had. We at one point we were at a consultation for Aurélien for his heart, and a doctor told us, yes, there is a study in the US, and it could be really a good thing to enroll Aurélien in the study. And so yeah, we say yes, of course. We signed the paper, and we went back in the car and realized that between the consultation and being in the car, we lost the paper. We don't really know in which study Aurélien is enrolled. And we believed it was not really optimal process. On the other hand, now that we were launching GEMS, we realized that for the principal investigator, it is really a challenge to pass his first ethics committee in his own institution. And after, the study has to pass the approval from the ethics committee from, for example, the doctor that presented the study in another institution. And after it ends in our hands, we signed it on the paper and we lost it. We believe that it was really not an optimal solution, neither for the participant, neither for the doctor, neither for the principal investigator. So we decided to create a solution that will satisfy the participant, that will allow the participant to always find back his consent, to actually really withdraw his consent, and to know what the investigator is doing with the data. And the idea is that the participant will be in a position where he can directly and dynamically consent with the principal investigator. So doing that, we create this thing, which is called the GEMS app, which is alive, which is fully functional, and that allows, that's functional it's multilingual. You can use it in French, English, Netherlands, Dutch, Spanish. You can do it for yourself or as a representative of a person in capacity. And there's a specific track to follow to gather the consent. There is a nice video that you have to watch done by Van den Heuvel, and she explained all the study. And we didn't want that you just sign the paper and go away without knowing what you do. And so at the core of this solution, we put a verification tool, meaning that if you want to consent, you have to demonstrate that you understand to what you consent and that you fully agree with this, with joining this study. So at the core of the system, there is this quiz, this verification tool. You have to answer correctly to 5 questions with different proposition of answers, and there's a different set of data. So if you miss it, if you fail the first time you try it, you can try it again and try it again until you are able to demonstrate that you understand sufficiently what you are really willing to join. When it's done, you are allowed to consent directly with the principal investigator of the study. In this case, is Professor Barkeris. As soon as it's done, it launches all the process on our side at the 101 Genome Foundation, and we send to you a saliva extraction kit that you receive by post, and it's free of charge when you use it, and after you send it back. Actually, there it's at the UZ Games laboratory, where the DNA, the biological DNA, it's extracted, and one part goes into the biobank, and one other part goes to the genomic cloud, and it's transformed into a digital data. When it's done, it's not finished for the participant. For the participant, at any time, the participant can go back to the portal, the MIDA, Manage Your Data portal, where he can every time, so every moment, find back his consent, withdraw his consent, actualize his data, provide additional data, know to what hand the data has been used, and exercise all the rights that the participant received thanks to the GDPR. And that's mainly another fully of other option that you can find on this portal. The idea is that the portal is bidirectional, meaning that the participant can decide to communicate more data if he wish to do so. The solution has been endorsed by several ethics committees, and this is actually this is a solution that's qualified now as a dynamic consent solution, and this is what is foreseen and recommended by the World Health Organization. So you understood it. The first part of the story, it's the birth of Aurélien. We went to the patient association because we knew nothing about the Marfan syndrome. We understood that there was something to explore and this quest for the modifier gene to launch. We created a legal body with the support of the King Baudouin Foundation. We created a biobank. We created the genomic cloud, and we created the consent management application that allow us to go back to the patient organization. And so with that, as of today, there are 140 people from all over the world that decide to join the GEMS study. They are coming from Belgium, France, Luxembourg, the Netherlands, Slovakia, Sweden, Austria, Spain, UK, Denmark, and the USA. And so we do some statistics, and we can see that the ranking is led by the French and the Spanish. And unfortunately, there's only one participant as of today from the UK. And it would be really brilliant if 10 more people could join, because it will allow us to reach 150. And so we will be able to continue our work. You can see that there is a discrepancy of the number of the participants between the men and the women, and also with the age. It's declining with the age of the participant. So today on our genomic cloud, we have 140 whole genome data of the participant. We also received exomes from another scientific group and panels from another scientific group. So as of today, we have 551 NGS data for Marfan patient that make us probably one of the largest genomic Marfan cohorts in the world, which we are really proud of. We are enrolled in the Genome for England, Genomics England initiative project, thanks to Dr. Lima Robert. And the GEMS project, you can see it's specifically recognized by the GEL. It's not as easy as we thought to access to the data on this side. And quite ironically, the first access request that we received regarding genome of Marfan patient came from the UK. So we really believe that what we do has a lot of meaning, even complement to the GEL initiative. So what we do with the data, mainly we work on this first one. We work on a tool that we call GemVap that allow to give a better level of classification of the pathogenicity of the variant in FBN1. This is a tool that's live. You can reach it here with this at gemvap.live, and you can use it, and you can try all the different variants that you can imagine on the FBN1 gene and try to see what are their pathogenicity predictor score. We have our own tool, which is GemVap, and we complement it with the alpha missense predictor solution. This is a tool that's already used in clinic, and for example, in the Loeys lab, to help them to accelerate the diagnosis of Marfan, which we are really proud of. On the top of it, as you understood it, what we need to do is to understand the interaction between the genes. So we created another AI tool that allows us to highlight the networks between the genes. And we did that with the team of Professor Tom Lennart, and it's a tool which is inside our genomic cloud, and that allows us to conduct directly on our data the identification of the networks between the gene involving FBN1 gene, which allow us now to check each time there is a publication about the modifiers, we can check if the modifier which is suggested appears or not in our database with our tool. So in the conclusion, what we wanted to do is to add a third path. There is a classical in vivo path with the mice and the fishes, and there is a more advanced approach, which is the in vitro path with the iPSCs and the organoid modelings. And what we wanted to facilitate is the in silico approach with this third part. So we created that. We have now 4 pillars, which is the financial and legal, the financial pillar with the King Baudouin Foundation. We have all the dimension with the legal and ethics, which is held by the GEMS app solution. We have all the biology with the biobank, and all the digital solution with the genomic cloud and the bioinformatic tools that we are using. So here, as I told you, we are at 140 participants as of today. It would be really brilliant if tonight we could have 10 more participants. And here there's a little video. I will try to play it. I hope it will work, because it will explain everything I showed you.
101 Genomes aims to advance genomic research dedicated to rare diseases. 101 Genomes focuses at first on Marfan syndrome. If you have Marfan syndrome, you can help everyone affected, and you can do it easily and cost-free. Let us explain. GEMS is a multicentric genomic study dedicated to Marfan syndrome, led by research groups at Antwerp University Hospital and Ghent University Hospital. 101 Genomes created a solution that enables you to join GEMS research wherever you are in the world. The goal of GEMS is to identify the protective genes able to counteract the main cardiovascular problems associated with Marfan syndrome. The GEMS design is to pool Marfan patients with severe cardiovascular problems and unaffected individuals harboring identical FBN1 variants in order to identify the genes that explain the difference. GEMS will lead to better patient counselling, better risk stratification, and development of new therapeutic approaches. Who can join? Every Marfan patient with a known FBN1 mutation can join, even if GEMS is particularly interested in a specific FBN1 mutation. How can you participate? To join, all you have to do is visit www.101gems.be, register and consent online to join GEMS. Once your consent is countersigned by GEMS investigator, you will receive a saliva collection kit at home. Follow the instructions provided in the box to take a saliva sample, then send it back to the laboratory free of charge. At the laboratory, GEMS researchers will isolate your DNA and sequence it so that your data can help in the quest for protective genes. Your biological samples will be preserved in the 101 Genomes Biobank to anticipate technological evolutions and to allow controls and verifications if necessary. The security and confidentiality of your data are at the core of our concerns. That's why our processes and storage infrastructure have been designed to guarantee optimum security and confidentiality. GEMS is supported at global level by the Marfan Foundation and in Europe by the national associations within the Marfan Europe network and by the Vassern Scientific Network. Don't wait any longer. Help yourself, help others, and help research advance today by joining GEMS. www.101gems.be. 101 Genomes Foundation. Genomics for Rare Diseases.
Et voilà, that's it from my side. I hope that it was interesting for you. And if you have any question, please, I will try to answer it.
Thank you so much, Romain. I expect that our research scientist, Jose. Yes, Jose's hand is raised.
I have several questions here. How can I get access to the data? I don't know if you said it already there, because I was a little bit overwhelmed with a lot of information here, which was very nice actually. And if we can get access, will it be possible to put the link in the chat so it will be easier to access that data? Or is not ready the data yet to analyze, and what type of data we should be able to access? We'll be able to access the whole genome sequencing, or we will be able only to get access to the final result? And also, my second question is that what techniques are you using apart from the whole genome sequencing? Are you using methylations as well?
Jose, I actually received your email and I replied to your email a month ago. And so thank you again for this more precise question. So at the time we have a data access committee process, we need to have specific research where you explain what you want to do with your access, and that will allow our data access committee to validate your access to the data. Currently, the way it works is that you ask your question, okay? You ask what you want to see in the genomes, and we revert to you with the answer, which is a bit the model that we see in the gel and that we had in the gel, and we are still waiting for the answer that you asked for the gel 3 years ago. But so if we can really work this way, but we need to have a research project that's recognized by an ethics committee from your side, and after we endorse it with our own DAC, and after you ask your question and we reply to you. In the future, we foresee to allow you to go directly on the whole genome. I don't know if you have tools that allow you to work directly on the whole genome, or if you prefer to work on the exome. This is a lot of questions that we have to ask to you. And I can tell you that we are not, a lot of people are asking us about the methylation. We are not working on the methylation at this stage. We believe that it's already a lot that we can provide the whole genome. And I believe that you will work, perhaps more working on exome, but we are able to provide that in HG38, if it makes sense for you.
Okay.
I can tell you that all the genomes are sequenced in Illumina 30X, if it's something that you need to know also. Yeah, but we can continue to have a more precise discussion.
Fantastic. Thank you.
Thank you.
I have one more question, but I'm going to let people first. I don't want to
but Jose, what really shows is that and it's really good for the Divine and I, is that at one point we say, okay, we gather that, we are funded by philanthropy, and we have this engagement that we gather 100 genomes, and we did it.
Yep.
And the idea that no people need to use it. So when we receive actually your request, Divine and I were really thrilled. We were just like, yes, it makes a lot of sense to create it. And even in the UK, where there is this wonderful 100,000 initiative, that was a kind of inspirational for us. So thank you again for sending us.
I mean, we have the database for many, many years because Dr. Char has been collecting it, and thank to the Marfan Trust for funding it as well, because we keep that database, otherwise it will disappear. But one of my questions is that if this variant is so common in the Marfans in the population, could not be that this variant is actually a polymorphism and another variant is actually causing the phenotype?
Exactly, and it's why we chose it, because it will help. But it's really a perfect question. It's really why we decided to go for this one. And I will not tell you more about that. We had the meeting. We had the fifth meeting of our scientific committee last Friday. And yes, this is really, really interesting, and it's why we chose this one. Indeed,
yep.
Yes.
Fantastic. Thank you.
I think Mr. Chandra, Aman Chandra, our next speaker, actually has a question for you.
Thank you. I think Anne Child had a hand up as well. I don't know how to put my hand up, so I'm sorry. So I'll put it in the chat box. But man, that's really interesting. I really enjoyed your talk. Can I ask you 2 questions, and I won't take any more of your time. One's very simple. You mentioned the mutation you're looking at in terms of cardiac manifestations, and then you're looking at people who've got that mutation who haven't got any physical features or any phenotype that you'd expect. How do you identify those patients without the clinical features? Are they in a database of family members, or how are they diagnosed with Marfan?
Excellent, really excellent question. So the first step, we went really the first level. We went by the clinical database from several hospitals involved in the VACERN project. Okay, and so we identified within the same family people harboring this variant and being affected, another member of the family being non-affected. This
so
is
relatives of the proband, relatives of the person who
exactly relative to the probands. That was the first approach. But when we launched the GEMS app, we had people coming from all over the world joining. And at one point, we had a really old man, okay, really old, not affected. And it appears that he was harboring the mutation that we were looking for. So the process, we had these two approaches, the approach by the centers and the approach directly with the patient. And it allows on both sides to identify really interesting participants, which we are really proud of.
Can I ask a second question? I was really interested, I know it's a small part of your talk about the dynamic consent.
Yeah.
And there's a whole research that you could develop on just on that process, because I would suggest that most consenting is limited, even in day-to-day clinical practice. I wonder if there's much scope for if one can use it in clinical practice, because I think it's a really interesting tool to demonstrate or to investigate how well people understand what they're being involved in, not just in research, but if you're having any treatment, we have to consent our patients. And I'm always wondering how good the consent is. And this dynamic consent, I wonder if there's scope for using it or investigating it or researching its validity in day-to-day clinical work. Because I've looked it up just now and I should know more about it, but I don't. And it all seems to be focused on research. As you used it for, but I wonder if there's scope for it to be used in a wider sense outside research, just in normal consenting.
Once again, a wonderful question. We are actually working on a publication with the research perspective, but indeed we have a lot of people coming to us asking us to duplicate this solution of dynamic consent in the context of research and to investigate how to use it in clinics. Okay, but we are actually, yesterday we had a workshop with the DG Connect of the European Commission, and it was quite a surprise to see, because we are feeling them with all the work that you are involving in this deployment of this dynamic consent application since 3 years now. And yeah, it was interesting at least to see what they did with all. So this is something that we will continue to work with. And
I think Dr. Child has a question.
Yeah.
Thank you for that, Romain. It's magnificent that you're putting so much effort into trying to improve the research scene. My first question is that as a clinician and the families who are attending this conference will realize that within a family, there can be tremendous variation. So you have a severely affected sibling and a not severely affected sibling. And it would seem to me that if you could study those and find out what protective genes the mildly affected sibling has, that would be a good approach. Are you asking people to enroll their siblings as well? For example, you can have height genes in a family and you find the taller child who has to grow more quickly and therefore the tissues are weaker, may be more severely affected, whereas the shorter child is protected from the connective tissue weakness by limiting the growth and the stress on the tissues. That's just one clinical observation. But if you could capture a sibling who was affected severely and a sibling who was affected not so severely, wouldn't it be interesting to study families rather than individual patients?
Yeah, to be really clear, this is the approach. This is the approach conducted by within the centers. So within the centers, the different centers are targeting the families and trying to identify within the families the siblings. This is how it works. But what we add to that is this individual approach by the participant, by people in the patient association, and we show them that they can be the actor of that. And actually doing that, it's what I explained with this French patient. We also detect families. So we have two different paths to approach the same participant group, actually. We have the one from the centers where we go directly and they know who are the siblings. And on the other side, we have the participant, like the people who are listening to us today, that want to join and do something that can help. And if they join, could also help us to identify potential siblings if they are. But it's not okay. This is the complementary approach that we have with the GEMS.
Good. So everyone who joins, if you could ask them, do they have siblings?
Yeah. No. Even if they don't have siblings, they are really welcome. Everybody can join. Okay. This is what we do at this stage, and after we do this filtering, and we go. And this is what the centers are doing.
Thank you very much. Okay. Back to you, Victoria.
Does anyone have any further questions for Romain?
I just would like to add something concerning the previous question on the app that we created. So I'm Ludivine, I'm Romain's wife, so I created one Genomes with him. Nice to meet you all. In the app that we created, we allow the participant to fill in their medical data if they want to, and also there is a genealogic tree, and so they can say if some people in their family, and if some siblings are affected or not. So the patient and the participant can also share this info if they want to with us. So that's what I wanted to add. Thank you.
Thank you so much for clarifying. Any further questions? Of course, if something occurs to you later, you can always email us. I don't know if it's time now to move on to thank you, Romain.
I see that there are some questions in the chat. I will try to answer to them. No, but I will try to answer to them. And otherwise, I will also let in the chat my email address. And so feel free to connect. And I will also put the link to use the GEMS app. And as I told you, it will be really wonderful if more people could join the research. It will really change our world. So, thank you.
Thank you so much. Thank you.